Hello dear students, you can see. Let's start our lesson. Today's topic is the mechanism of passive transport, and today's goal of our lesson explain the mechanism of passive transport and our success criteria. Describe the type of passive transport and explain the mechanism of passive transport. And now let's continue our lesson with some essential questions. Let's move and go to whiteboard.
Cells, whether in an animal's body or living independently, are surrounded by a watery environment. What keeps a cell's contents, which is mostly water, from escaping into its surroundings is a clear envelope called the plasma membrane. The membrane itself is too thin to be seen with a light microscope. What we are actually seeing is the boundary between the cell's cytoplasm and the surrounding fluid. That a membrane is present becomes evident as this amoeba is squeezed under a cover glass, rupturing its plasma membrane and spilling its contents into the surrounding water. The membrane not only contains and protects, it's also a gateway for molecular traffic. One kind of molecular traffic that has a very immediate effect is the movement of water molecules through the membrane. Water molecules are in constant motion, causing particles suspended in water to dance. The particles are responding to the bombardment of speeding water molecules.
To learn the mechanism of passive transport, we need first to understand clearly what is the specific of the transport in the living organisms, and second, to know the details of the cell membrane structure. Let's talk about the transport in the living organisms. We have several transport systems in our bodies, such as circular and respiratory, but in each case we talk about moving from one cell to another one. And as you know, cells have boundaries, such as cell membrane and probably cell wall, and substances go across these barriers. So, in terms of living bodies transport, we should imagine the cell membrane structure before. The cell membrane structure. The cell surface is extremely thin, about 7 nanometers. However, at very high magnifications, it can be seen to have 3 layers, described as tri-laminar appearance. The membrane is partially permeable and controls exchange between the cell and their environment. Like all other cellular membranes, plasma membrane consists of lipids and proteins. The fundamental structure of the membrane is the phospholipid bilayer, which forms a stable barrier between inside and the outside, inside and the outside of the cell. Let's talk about the phospholipid bilayer structure. If you look at the cell membrane, we can see that phospholipids have 2 main parts, head and 2 tails. The head loves water. We call it hydrophilic. And the tails hate water, in opposite hydrophobic. The water-hating tails are on the interior of the cell membrane, whereas the water-loving heads point outwards, either the cytoplasm or maybe the fluid that surrounds the cells. The structure of the cell membrane is extremely important. The fatty acids of tails are poorly soluble in water, and they form a stable barrier between cell and their environment and separate the cell from its surroundings. Hydrophilic heads, however, gives ability to interact the cell with each other and with their environment. And now let us look at the video about the cell membrane structure.
The plasma membrane is made up of several organic molecules. I will concentrate on two of them in this presentation. The first molecule is a lipid. It is called a phospholipid because instead of the usual 3 fatty acid chains, there are only 2. The third bonding position is with a phosphate group. The phospholipid molecule is divided into a head region and a tail region. The head region is the glycerol and phosphate group. The tail region is the fatty acid chains. The head is hydrophilic, attracted to water. The tail is hydrophobic, repelled by water. It is this property which allows for the unique molecular alignment that makes the plasma membrane. If a droplet of phospholipid was put into water, a mycele would form. A mycele is a sphere with the hydrophilic heads on the surface, the tails inside, away from the water. Our bodies are made up of approximately 60% water and trillions of cells. The water can be found inside the cells, outside the cells, and in the bloodstream. The location of water and the phospholipid, hydrophilic, and hydrophobic ends enable the membrane to form a bilayer of lipids. The currently accepted model of the cell membrane was developed by Singer and Nicholson in 1972 at the University of California, San Diego. Pictured here, the plasma membrane looks like a sea of lipid with icebergs floating in. The membrane is very flexible and has a fluid motion. This design of the plasma membrane is called the fluid mosaic model. Quite often, these proteins have sugar molecules attached to them, forming glycoproteins. These molecules extend from the membrane surface and act as an identification marker. The proteins are used by the immune system to identify a cell as being self, or foreign called non-self. Each individual has their own biochemical makeup. In other words, each human being has their own set of cellular proteins. But if a cell comes into the body that has a different set of proteins, whether it is a bacteria or an organ transplant, the immune system may identify it as self or as non-self and attack. In order for the cell to survive, many different materials must be able to cross the plasma membrane. When water passes through a cell's membrane, the type of diffusion is called osmosis. When solute particles pass through a cell's membrane, the type of diffusion is called dialysis. Before looking at this movement through the membrane, it is important to understand the movement of molecules in general. Atoms and molecules are in constant motion. This random motion will disrupt the cluster rather than maintain it. This motion is referred to as Brownian movement. This motion is always present in some degree, in gases, in liquids, and even in solids.
Substances can enter or leave a cell. It's a passive and active. A passive is a simple diffusion, facilitated diffusion, and osmosis. It's for water only. And active is for molecules and for particles. And one very important thing about the passive transport. Passive transport mechanism use no external energy source to bring about diffusion of a substances across a membrane. Let's look. It does not energy. And now several important definitions of our topics. First one is diffusion. Diffusion is the process by which molecules spread from areas of high concentration to areas of low concentration of substances. When the molecules are even throughout a space, it is called equilibrium. Next definition is a concentration gradient. It's a difference between concentration in a space. Next one is osmosis. Osmosis is the diffusion of water across the membrane from areas of high concentration to areas of low concentration of this substance. And next one is a facilitated diffusion. Facilitated diffusion is a movement of specific molecules down concentration gradient, passing through a membrane via a specific carrier proteins. And now let us talk about the types of passive transport that present our students. And now we are going to work in groups. Each group will discuss only one type of the passive transport. And after each group will present their job. Please come to this table and choose the label of types of passive transport.
Simple diffusion. Osmosis lab. Osmosis.
Oscillating diffusion.
Okay, let's start.
Osmosis, the net of movement of water molecules from a region of higher water potential, diluted solution, to a region of lower water potential, concentrated solution, through a partial permeable membrane. Osmosis is a special example of diffusion.
Diffusion and osmosis are both passive. Energy from ATP is not used. A partially permeable membrane is a barrier permits the passage of some substances, but others. It allows the passage of the solvent molecules, but not some of the larger solution molecules.
If we take two cells, such as these two, water will move in this direction, from area with higher concentration to area with a lower concentration of water.
Water entering the cell will make it swell up, and unless the extra water is expelled in some way, the cell will burst.
Conversely, if the cells are surrounded by a solution which is more concentrated than the cytoplasm, water will pass out of the cell by osmosis, and the cell will shrink.
Excessive uptake or loss of water by osmosis may damage cells.
For this reason, it is very important that the cells in animal bodies are surrounded by a liquid which has the same concentration as the liquid inside the cells.
In vertebrates, the concentration of blood is monitored by brain and adjusted by the kidneys.
By keeping the blood concentration within narrow limits, the concentration of the tissue fluid remains more or less constant, and the cells aren't bloated by taking in too much water or dehydrated by losing too much.
Some major examples of osmosis are absorption of water by plant roots,
Reabsorption of water by the proximal and distal convulsed tubules of the nephron.
reabsorption of tissue fluid into the venule ends of the blood capillaries.
Absorption of water by the alimentary canal, stomach, small intestine, and the colon. Plant cells in hypotonic solution cell have lower water potential. The plant cells gain water by osmosis. The vacuole and cytoplasm increase in volume. The cell membrane is pushed harder against the cell wall, causing it to stretch a little. The plant tissue becomes stiffer. Target.
Plant cells in hypertonic solution. Cells have higher water potential. The plant cell loses water by osmosis. As vacuole and cytoplasm decrease in volume, the cell shrinks away from the cell wall. Shrinkage stops when the cell sap is at the same concentration as the external solution. The plant tissue becomes facet. It has shrunk slightly, may go on to become plasmolyst. Turgor. Turgor is the pressure of the swollen cell contents against the cell wall when the external solution more dilute than the cell sap of the vacuole.
Role of turgor in plants. Mechanical support for soft non-woody tissue. Change in shape of guard cells forming the stomatal opening between them. Enlargement of young immature plant cells to mature size.
Let's say that I have this green container, and inside this green container I have some air molecules. Now the air molecules, we assume that there's some temperature, there's some average kinetic energy to them, but they're all going to have different velocities, they're all going to be bumping around in different ways. Now the way that I've drawn it, you might notice something. On the left-hand side, and I'll just draw an imaginary line here. This line has no real, I guess you could say, structural significance. It's not like it's actually dividing it. I'm just using it to visualize the left and right-hand sides. You see on the left-hand side, I have a higher concentration of my molecules. Higher concentration. And how do you measure concentration? Well, the number of molecules per, well, the really way you should do it is unit volume, but we're looking at a cross-section here. If I were to take a section that large, look, I got 4 molecules here. It looks like I have about 3 to 5 molecules per section around that size. Well, if I took that size section on this side, I'm getting 1 or maybe 2 molecules. And I'm not going to get too precise, but it's clear that I have a higher concentration here. I have more molecules the way I've drawn it per unit area, but if we were thinking in 3 dimensions per unit volume, than we have on the right-hand side. So we have a higher concentration on the left. We have a lower concentration on the right. Lower concentration on the right. And when you have this situation where you have a higher concentration and then a lower concentration, we call this a concentration gradient. The concentration is changing from high to low, and so we call this a concentration gradient. Now, what do we think is going to happen? Let's say this is what our situation is right when we look at it. What do we think is going to happen? Well, all of these particles are all going to be bouncing around. Things can move from the left to the right. Things can move from the right to the left. But we have more particles on the left that are likely to move to the right than we have particles on the right that are likely to move to the left. Remember, they're all moving in different directions with these random velocities, but I have more on this side, and they're all bouncing around. So in any given moment, when we have this higher concentration on the left, I have a higher chance that I'm going to have stuff go from the left to the right, than I do from the right to the left. And so as time goes on, it's going to look something like this. If we let this system stabilize, for a while, it should look like this. And let me see if I can do a good job drawing it. And I'll just draw the molecules. I won't draw their actual velocity vectors. So if we wait a while, how many molecules did I have? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So now I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So if we let enough time go by, we see that we should no longer have a concentration gradient, that the concentration should be fairly uniform over time. So even if I were to draw that imaginary dashed line, I should have the same concentration on both sides. So I no longer have a gradient. I have no gradient. And once again, there's nothing magical here. It's not like the molecule said, oh, we are less concentrated over there. I somehow have to know to move there. You just have to think that these are all just randomly bouncing around. And if you have a higher concentration on the left, there's a higher chance that you have bounces or you have things moving from the left to the right as you do from the right to left. Even in this situation, things are still going to be moving from left to right and right to left. But now that you have the same number on either side, in any given moment, in any given period of time, you have an equal probability of things moving from the left to the right as you do from the right to the left. So you're getting to kind of this equilibrium situation. Sure, in a given, if you take a certain unit of time, maybe that one moves from the left to the right, that one moves from the left to the right, that one moves from the left to the right. But since you have equal concentrations on both sides, you're just as likely to have the same number move from right to left. And I only did this with 20 particles, which is a little bit of an artificially low number. If we're actually talking about concentrations of air molecules, or as we'll see when we think about cellular membranes, if we think about different types of molecules that might be in an aqueous solution, we're talking about way more than 20 molecules. And so you really do think in terms of probabilistic large numbers. Well, hey, for the probability of something move from the left to the right is the same as the right to left, and so you're going to have the stability. Right here, there's a much higher probability in any given moment of something moving from left to right than right right to left, and that's why you see things moving from high concentration to low concentration. Or another way to think about it, what we just observed here is we saw things diffusing down their concentration gradient. So this process that we just described, this is diffusion. This is diffusion. And as we study different types of systems, we'll see that this is actually very important to biological systems and even chemical systems, because this doesn't require any extra energy to move the molecules from here to there. This is going to happen probabilistically. It's going to happen naturally. And once again, no magic. Just more stuff here, higher chance moving from left to right than moving from right to left. And I really want to make that point clear. You can still move from right to left. For example, you might have this character, maybe instead of moving in that direction, completely possible that he goes from right to left. It's not like everything is moving from left to right, but you have a higher chance you're going to have more things moving from left to right. So that guy could move in that direction because there's just more stuff here, and they're all bouncing around in all different random directions.
Diffusion is a net passive movement of particles from a region in which they are in high concentration to regions of lower concentration.
It continues until the concentration of substances is uniform throughout.
Here we see green water particles and red particles of another substance. And this is there is a lower gradient concentration. It means that shallow concentration gradient means a slower rate of diffusion.
There's green color is water particles and red color is another substance. Steeper concentration gradient means a faster rate of diffusion.
High diffusion rate, short distance, large surface area, big concentration difference, fixed law. High temperatures increase diffusion, large molecules slow diffusion.
Fixed law.
Number of proteins channels will affect rate of diffusion too. Temperature too. More heat means more kinetic energy.
Rate of diffusion equal to surface area multiplied by concentration gradient and divided to diffusion distance.
Some major examples of diffusion in biology. Gas exchange at the alveoli. Oxygen from air to blood. Carbon dioxide from blood to air. Gas exchange for photosynthesis. Carbon dioxide from air to leaf. Oxygen from leaf to air.
Gas exchange for respiration. Oxygen from blood to tissue cells. Carbon dioxide in opposite direction. Transfer of transmitted substances. Acetylcholine from presynaptic to postsynaptic membrane at the synapse.
Lab for diffusion demonstration.
In this demonstration, we will make observation of diffusion of a semi-permeable membrane, and we will be able to compare this observation to the function of the cell membrane.
Iodine and starch are used in this lab because iodine is a known indicator for starch. Iodine will change colors in the presence of starch. Materials needed: dialysis tube, here, iodine, starch, 2 large clear beakers, and distilled water. First of all, we fill a dialysis tube from 1 spoon of starch and half cup of distilled water. First of all, we should fix the dialysis tube. We should tie the dialysis tube closed, like here. We fill a dialysis tube with 1 spoon of starch and a half cup of distilled water. After, we should place the dialysis tube and fix it. Like this.
And put iodine solution inside this. What do you think will happen when we put the baggie and the iodine water mixture.
After about 15 minutes, the iodine will have seeped into the dialysis tube, turning the bottom part of the dialysis tube starch a purplish black color.
As a result, the liquid inside the dialysis tube goes blue with the iodine solution.
The blue color is characteristic of the reaction which takes place between starch and iodine and is used as a test for starch. The results show that glucose molecules have passed through the dialysis tube into the water, but the starch molecules haven't moved out the dialysis tube. This is what we would expect if the dialysis tubing was partially permeable on the basis of its pore size.
Starch molecules are very large, probably cannot get through the pores.
Osmosis is the movement of water molecules across a semi-permeable membrane from an area of greater concentration to an area of lesser concentration. Here we have red blood cells in solution. The red blood cell has a semi-permeable membrane. Tonicity is the measure of the osmotic pressure of two solutions separated by a semi-permeable membrane. It is commonly used when describing the response of cells immersed in an external solution. Like osmotic pressure, tonicity is influenced only by solutes, such as sugar, that cannot cross the membrane. There are three classifications of tonicity that one solution can have relative to another. Those three classifications are isotonic, hypertonic, and hypotonic. If a solution is hypotonic, there is a greater concentration of solute within the cell than there is in the surrounding solution. As a result, water molecules enter the cell. As the water molecules continue to enter the cell becomes larger and larger. If water molecules continue to diffuse into the cell, it will cause the cell to swell up to the point that lysis or rupture may occur. In this demonstration, dialysis tubing and a sugar solution are used to demonstrate a hypotonic solution. First, a 3 centimeter length of dialysis tubing was cut. The tubing was briefly soaked in water, then rubbed between the fingers to open the tubing. One end of the tubing was twisted, doubled back, and secured with an orthodontic rubber band to prevent leaking. The tubing was then filled with a concentrated sugar solution and sugar granules. The second end of the tubing was also twisted, making sure that no air molecules were in the cell. An orthodontic rubber band was used to secure the second end as well. The completed cell was weighed on a digital balance and then was gently placed in a beaker of tap water. After 30 minutes, the cell was removed from the beaker, gently patted dry, and again weighed on the digital balance. As you can see, there was a significant gain in weight from the water molecules entering the cell. If a solution is hypertonic, there is a greater concentration of solute outside the cell than there is inside the cell. As a result, water leaves the cell and the cell shrinks. In this demonstration, a 3 centimeter length of dialysis tubing and concentrated sugar water again were used. The cell was created as before, except this time tap water was poured into the dialysis tubing cell. After both ends were secure, the cell was weighed on the digital balance. The cell was then placed in a beaker of concentrated sugar solution. After approximately 30 minutes, the cell was removed from the sugar water solution and again weighed on the digital balance. As you can see, the cell lost a significant amount of weight and has become very flaccid or lacking in firmness. When a cell shrinks, It is said to be crenate. If a solution is isotonic, there is an equal concentration of solute both inside and outside of the cell. Although water molecules continuously move into the cell and out of the cell, the concentration on both sides of the semi-permeable membrane remain the same. In this demonstration, a cell was again formed from the dialysis tubing and was filled with tap water. After both ends of the cell were securely fastened, this cell also was weighed on a digital balance. The cell was placed in a beaker of tap water for half an hour. As you can see, the weight of the cell remained constant.
The question for today is how potato cells react in different solutions and what will happen in hypertonic and hypotonic solutions. And we expect that potato in hypertonic solution will lose mass, but on the other hand, the hypertonic solution